All right, thank you guys for your patience in waiting for the cell cycle and mitosis video. So let's get to the notes. So before we get into the more kind of complicated mechanisms of reproduction or replication in eukaryotic cells, I wanted to talk really quickly about what happens in asexually reproducing organisms. The one example we're going to look at is bacteria. So they're unicellular. So basically for them to reproduce, they just need to divide their cells. And when they do this, they produce clones. So they'll make an exact copy of their single chromosome, which is what you see happening in step 1. The cell replicates its DNA, then it grows and elongates, and then pinches off basically to form 2 new cells. This very simple form of cell division is called binary fission. Literally, it means, binary means 2, fission means to split. So literally, it's splitting into 2. The process becomes much more complicated in eukaryotes because we have a nucleus, we have multiple chromosomes. So we'll be looking at that. I'm also going to be pointing out throughout this lesson, as well as our next lesson or two, we're going to look at sources of genetic variation, because our next unit is going to start getting into genetics, and then after that we're going to get into evolution, where these concepts are really important. So asexual reproduction gives you no additional genetic variation. So let's look, now that we have looked at cell division in prokaryotes, let's look at the differences in DNA between prokaryotes and eukaryotes. So prokaryotes have a single circular chromosome, usually around a few thousand base pairs, although they can get a little larger. And they're replicated and distributed during binary fission. They don't have a very complex structure to them, so they're relatively simple to replicate and then package into a new cell. Eukaryotic cells, on the other hand, have multiple linear chromosomes. And this number can be anywhere from a few chromosomes, I believe, if I remember correctly, fruit flies have like 4, to some plants which can have over 100 chromosomes. They're all linear, and if you've ever had like a bunch of string or yarn or thread and or necklaces even, and you've been trying to untangle them, you'll know that when they're linear, it's a lot harder to untangle them. So your cells do have mechanisms to kind of keep your DNA packaged and organized, and that is basically chromosome packing, which happens with the help of proteins called histones. Your DNA for a eukaryotic cell is replicated during S phase, which is part of the cell cycle, and if you don't remember it, we're going to be talking about that in a moment. And then it's actually distributed during mitosis. So mitosis is strictly the nuclear division part. So what happens when you have mitosis about to happen is your chromosomes condense. What basically happens is your DNA double helix gets wrapped around histones, which are proteins that are involved in the packaging of DNA, and then those kind of strings of histone wrapped DNA coil up, which then also coil up, and you're basically making coils of coils of coils, until you finally get this very compact structure called a chromosome. Now, in this state, the cell is really not able to access the DNA. So if you're wondering, why don't our cells just always keep their DNA in the form of chromosomes? Why do they ever unwind it? Well, if they kept their DNA compacted all the time, they would never be able to access genes and never be able to produce proteins. So while DNA is compacted for moving it around during mitosis, most of the time it is unwound a lot more, although there are segments of DNA that might be wrapped up around histones and kind of condensed. It might stay in, I do not know why my pen is deciding not to work. We'll try and deal with the pen issue. But there are some parts of DNA, for example, if you have genes that are just not active in a particular cell type, they may be condensed long term. So let's go through the cell cycle. The entire purpose of having cell division, having a life cycle for a cell, is it controls the production of new cells. You need new cells in some cases if you're talking about single-celled creatures for reproduction. Mostly we use it for growth and regeneration. So if you damage tissue, if you damage cells, they can in some cases be replaced by new cells. So you have this diagram on your printed notes with some gaps. Let's go over some of the highlights. I'm not going to go over this in as much detail as you might like because I'm hoping that you'll jog some of your memories from freshman bio. So the cell cycle, we usually say it begins after mitosis. So the first stage of the cell cycle is called G1. I think of the G as standing for growth because during both G phases the cell grows, but technically it was named gap 1 because the scientists who were studying it didn't think anything important was happening. So usually after cell division, a cell might be a little smaller than its normal size, and so during G1 phase there's growth. And then the cell is able to perform its normal functions. Now, notice that there are stars located around here. These stars represent checkpoints. At a checkpoint, a cell is going to make a decision about whether to move on to the next stage in the cell cycle, whether it needs to do something to prepare for the next stage in the cell cycle, or even if it is just catastrophically unable to perform cell division, and then, in that case, what happens is the cell can undergo apoptosis. So at this point, a cell has a decision to make, and based on, various factors, growth factors in its environment, internal signaling, the type of cell it is, it may either exit the cell cycle, go into something called G0, which is cell cycle arrest, or it can go on to S phase, which is the next phase in the cycle. Some cells, when they go into G0, they're going to stay in that phase for the rest of your life. Other cells can actually re-enter the cell cycle when they receive the proper signals. So S phase stands for synthesis. That is when you replicate your entire genome. So every single chromosome is replicated. And you'll notice here we have a single chromosome, but after S phase, you have these kind of joined chromosomes. Those are, we'll talk about what they're called in a moment. Then during gap 2 or G2, the cell grows, you produce a lot more organelles, so that way you're prepared for division. There is a checkpoint at G2, and that is the cell checking, is all my DNA copied without mistakes? Do I have enough material for mitosis? And if the answer to those questions is yes, then the cell will enter mitosis. During mitosis, the chromosomes, the two copies of each chromosome, will be pulled apart to opposite ends of the cell. And there's also a checkpoint here. At this point, if you don't have proper attachment of the chromosomes to structures in the cell, you'll stop mitosis until you can fix that. And then finally, after mitosis, or at the end of mitosis, you have cytokinesis happening, which is literally splitting the cell, the two new cells apart, and then the cells that are produced are called daughter cells. So everything except for mitosis and cytokinesis is called interphase. The cell actually spends most of its time in interphase and relatively little time in mitosis. We probably give you the wrong idea about that because we tend to spend so much time on mitosis, but that's just because there's a lot of stuff going on during mitosis. So let's look at the structure of a chromosome so that way we will be able to use some terminology that you may not remember. So a single unduplicated chromosome looks like this guy right here. Notice it's got a centromere kind of towards the center. That's an area of the chromosome that is specialized to attach to the centromere of another chromosome. It's usually like repeating sections of DNA that don't actually code for genes. The parts of the chromosome that stick off from the centromere are called arms, and you don't need to know a whole lot of details about them. When a chromosome is duplicated, after you have S phase and synthesis, you have two identical chromosomes. They are exact copies of each other attached at the centromere, and those are called sister chromatids. So let's talk about something important that is found at the centromere. So centromeres are where sister chromatids attach to each other. But later on, we're going to have some structures attached at the centromeres, and they're going to attach to something called the kinetochore. If you remember your physics, what does kinetic mean? Well, kinetic means movement. So the kinetochore is the part of the chromosome that's going to allow for movement, and that's the point at which the basically fibers of the protein spindle, if you remember what that is, will attach and start to pull these chromosomes around. So that's where the movement's going to happen. You can see a little bit of detail here where you basically have a kinetochore attached to a microtubule. The microtubule gets fed through and it actually starts getting disassembled there. But we'll talk about that in more detail later. A few other important structures for mitosis are the centrosomes, where you have a pair in animal cells of centrioles, which are these structures pictured here. And that creates the other important structure, the protein spindle. In plants, this is called the MTOC, or the microtubule organizing center, just in case you see something on mitosis in plants on the test. The protein spindle is this network of microtubules that spread throughout the cell. They have two major jobs. One, you're going to have some of them that are going to connect to the kinetochores of those chromosomes and work to pull them apart. Then the other fibers of the spindle that are formed, they basically help push against each other to start elongating the cell, making it longer. So that way, when you have your chromosomes separated, you can split into two different cells. So you don't necessarily need to have like the names of all the phases memorized, but you do need to remember what's happening and when. I am going to use the names of the phases. And it's helpful for you to know them. That way, if you get asked a question on an FRQ and they don't tell you what anaphase or metaphase is, you won't be so lost. But mitosis is division of the nucleus. So it's divvying up your chromosomes to make sure that each new cell gets a full copy of every bit of DNA. Because if you're missing some DNA, that could be, that will likely be terminal for that cell. It's going to apoptose because it doesn't have all the genes it needs to function. Everything else that's split up during cell division is going to be split during cytokinesis. So let's look at important moments in mitosis. First thing that happens is you have the chromosomes attach at their kinetochores to the spindle fibers. That part is really important because without that, you can't move the chromosomes around. Remember, these are molecules. They don't walk on their own, and they don't have any sort of like motor proteins on them or anything to move them around. So they're going to get yanked around by the spindle fibers. By the way, this is what happens during prophase. Other thing that happens during prophase is the nuclear membrane kind of breaks down, and that kind of like clears the slate so that you have room to perform the rest of mitosis. And sorry, I don't know why the pen is doing this. I think my computer's having issues. Oh well, we'll live. Alignment is what happens next, and that is metaphase. Hopefully, meta, means middle. And what you can see is that the chromosomes all line up at the middle of the cell. It's an imaginary line called the metaphase plate. It doesn't actually, it's not like differentiated by anything physical. It's just the center of the cell where the chromosomes are going to line up. They're going to line up at the center of the cell. That way it's easier to pull them apart, and this just is an organizational thing. And then there's separation. So here you see a microscope image of a cell in anaphase. What you may not be able to see as clearly is that what's happening is that, okay, let's see if this will work now, is that chromosomes or chromatids are actually getting pulled towards opposite poles of the cell. By the way, once a pair of sister chromatids gets separated into those two separate chromosomes, they are called chromosomes again. I know it's confusing. If I think of a good way to remember that, I will let you know. So what ends up happening is the chromosomes, the individual chromosomes now, are pulled by their centromeres to opposite ends of the cell. When you have those chromosomes at the opposite ends of the cell, they can then basically be separated and the cell can divide. So that is cytokinesis. Cytokinesis literally means cell moving. It starts to happen during the last part of mitosis, which is called telophase. And during telophase, that's when you basically reverse all the changes you had in prophase. So your chromosomes unwind, your nucleus reforms, all of that. In animals, what ends up happening is there's this ring of microtubules around the center of the cell. They basically tighten like a belt until they squeeze the cells in two. In plants, a lot of vesicles are made. They line up towards the center of the cell, of the old cell, and when they join together, they form what's called a cell plate, and that is an actual physically visible structure. And then once they join with kind of the ends, where there's already cell wall and cell membrane, they form a brand new boundary between the cells. So the cell cycle is very complex, and like I said, there are checkpoints. So there are mechanisms to control the cell cycle, how fast you go through it, whether you move on to the next stage. So checkpoints are basically places where the cell pauses and checks whether conditions are favorable to allow the cell to progress to the next stage. So there are three major checkpoints we're going to look at. The first one is the G1 checkpoint, and that's basically, are we going to divide? So you can think of this checkpoint as, if I could write, it's supposed to say to divide or not to divide. I have the feeling it's going to show up like right when I'm switching to the next slide. So this decides whether a cell is going to enter G0 or whether it's going to move on to S phase. So if your DNA is nice and undamaged, you'll likely move on. If there are enough resources to support a new cell, then you can pass this checkpoint. If there's some signal, some external signal like a growth factor that is trying to stimulate the cell to divide, that can help move you past this checkpoint. The G2 checkpoint is basically, are we going to divide? Should we enter mitosis? So at that point, the cell's going to check, is there sufficient organelles? Do we have enough of all the proteins we need in the cytoplasm to separate into two cells? Have we replicated all of our chromosomes correctly? And if not, then the cell will pause at this point in the cell cycle and attempt to fix the problems. If the problems aren't fixable, then it'll basically trigger apoptosis. If the problems are fixable, the cell will resolve that, recheck, and then once it passes this checkpoint, it moves into the M checkpoint. At this point, this is during metaphase. and prophase. And that is going to be a check to see whether chromosomes are attached to the protein spindle. So it's not, are all chromosomes attached to the right kinetochore? It's, are all chromosomes attached to the protein spindle? And then whether or not the chromosomes will be able to be divided properly. If you don't have sister chromatids attached to spindle fibers from opposite ends of the cell, you pause, reattach, and if that doesn't work, then you go into apoptosis. So this entire process depends on a few specific molecules, cyclins and CDKs. Cyclins are so named because they control the cycle. CDKs are cyclin-dependent kinases. So since they're kinases, they are going to phosphorylate other proteins. And so they are going to phosphorylate proteins involved in moving on to the next stage of the cell cycle and activate them. They are inactive until they bind to a specific cyclin. So they can be in an inactive state or an active state. Each CDK has a particular cyclin it binds with. There are some simpler cells that only have one CDK, one cyclin. There are others, like I think in our cells, there's a good 5 that are active to control the cell cycle. So cyclin each cyclin only exists in the cell for a certain part of the cell cycle, but the CDK is constantly available in the cell. So whether or not you move on to the next part of the cell cycle depends on whether or not the appropriate cyclin is present. When the appropriate cyclin is present, then you will move on to the next stage of the cell cycle because it will turn on the CDK and allow the CDK to function and phosphorylate proteins. After the CDK is done moving you on to the next stage of the cell cycle, then cyclin's broken down by enzymes until you get to the next point, the same point in the next cell cycle. So one of the well-studied examples of this happening is mitosis promoting factor, or MPF. This is what ends up passing you through the G2 checkpoint, and it's named the way it is because it promotes mitosis. So when this is available in the cell, then it's going to trigger the cell to move on to mitosis. So there's a CDK that binds to a cyclin, and the protein that's produced is, or the resulting protein is called MPF. So what you can see here at the top is this is MPF activity and this is cyclin concentration. Notice when the concentration of cyclin is highest, that's when the MPF activity is highest. And it's going to be highest basically right when you hit mitosis, because you need high enough levels of MPF in order to move on to mitosis. What you'll see down here is just showing you the cyclin and CDK activity. So the CDK is constantly there. The cyclin is produced during G2. It starts to build up in concentration, and then it binds to the CDK to form MPF, the mitosis promoting factor. And then finally, once you've gone through mitosis, the cyclin is degraded, the CDK sticks around, and you're waiting for basically the next time you get to G2 in the cell cycle. So growth factors, and this is our overlap between our previous major topic and this one, growth factors are signaling molecules. They come to a cell, they bind to a receptor, and then they're going to initiate a specific signal transduction pathway, which is then going to basically trigger you to move on past the G1 checkpoint and initiate cell division. A lot of these are abbreviated with something ending in GF, which stands for growth factor. So for example, this PDGF is platelet-derived growth factor. You don't need to know where it got its name or what it is. You just need to know that since it's a growth factor, it's going to promote basically prepping for mitosis, because it's going to promote you having cell division happen. Now, there are a number of cells that are involved in these signals of whether or not to move on to the next part of the cell cycle, or whether or not to apoptose, like I've talked about a couple of times. A lot of these genes are called oncogenes because when they get mutated, when they are damaged, they usually will lead to cancer. So one of these is an important tumor suppressor gene called P53. A tumor suppressor gene is so called because when it is functioning properly, it suppresses it suppresses cancers. So it's one of your natural mechanisms to try and make sure you don't get cancer. So when there are cells that are damaged, their DNA is damaged, there's something wrong with the cell, then P53 is activated and it pauses the cell cycle. If at that point things can be repaired, they will be repaired, and then the cell will continue to go through the cell cycle. If the damage is too great, it triggers apoptosis. This is where a line from Star Trek really kind of comes in handy. The needs of the many outweigh the needs of the few. Basically, the cell will sacrifice itself to make sure the organism as a whole is able to survive. However, when you have mutated P53, it is not able to arrest the cell cycle, and those cells can then grow and continue to grow without limit. And if those cells just keep growing without limit, they become cancerous. And then non-dividing cells, there are a couple of examples of these. Neurons and muscle cells usually enter G0 and then they don't start dividing again. There aren't a lot of actively dividing populations of neurons, and I don't believe muscle cells reproduce at all past a certain point. There are other cells which are like progenitor cells or stem cells. They will enter G0, and then once you get a signal that more of that cell type is needed, then they'll re-enter the cell cycle and reproduce. All right, that's it. Sorry for the cursor issues, and hopefully that can be resolved before the next video.